Well, let's get this part that's less interesting over. Hi, welcome. Hi. Thank you so much for coming out in this kind of crummy weather. I saw hail or possibly freezing rain or possibly rain. Anyhow, it's very blustery, and I'm glad you came. How many people are from Harvard? Oh, good. So all the other people are not. Well, thank you very much for the Harvard people for coming, and especially the non-Harvard people. My name is Melissa Franklin. I realized I should introduce myself, and I'm a physicist involved with physics. So I want to introduce Susan Mango. I'm so happy she, after a year and a half, agreed to give this lecture. And Susan's in the biology department, one of the biology departments. She actually was a Harvard undergrad, and then for some reason went to Princeton and to do her PhD. And after that, she was at the University of Wisconsin as a postdoctoral fellow, and then moved out to Salt Lake City, University of Utah, to work as an assistant professor and possibly associate professor. Then, just at that moment, she got a MacArthur grant. And if I'd known that, I would have had her take me out for dinner for her work. And then just at that moment, she was already moving. Actually, she got the notification while she was waiting at the airport to move to Harvard, back to Harvard. So we have her back here. And she works on C. elegans, this kind of wormy thing. And I think what she tries to do is figure out how cells decide to become the final thing they become. But I think what's really exciting for me today is that I've heard a lot about this new class called Life Sciences 1A, which has been around for about 6 or 7 years, and every Harvard undergrad has to take it. And I've heard wonderful things about it, but I've never actually gone into it because it's a little scary. And so Susan is going to share with us not a full 90-minute lecture, but a part of that, a normal lecture in this very interesting introductory life science course. So I hope you'll enjoy it. I'm really looking forward to it, and maybe I'll become a biologist. So here's Susan Mango.
Well, thank you, Melissa. Is there any way I can convince you guys to come closer? We're sort of an intimate group, and it would be a whole lot more fun if you moved a little more close down. We could actually have a back and forth conversation. Yeah, great. Say what? I can't hear you.
I got to get up in the front.
Yeah, there you go. It's very interesting to see in the class, actually, who sits in the first sort of 4 to 5 rows and then who's sitting in the back. The guys, reading Facebook are sitting up in the back. And then you have the ones down here. The other question I had was, how many of you remember a little bit of your college science, a little chemistry, a little biology? And how many have nothing to do with science, no idea? If I say a proton or a protein, they could be the same thing as far as you're concerned. Okay, so what I'll do, this is a part of a class that I gave last fall. And as Melissa said, it's given to freshmen. They've just arrived at Harvard. This is their first science class. A lot of them are kind of thinking they might be pre-med, but maybe they want to go into chemistry. They haven't quite figured it out. And so this is the class that many of them take. And they walk into this room of 500 kids. It's given next door. So what I'm going to do is, on the one hand, I'm going to give you the lecture pretty much the way we give it to the students, because I thought you might get a kick out of seeing what a freshman learns in that first semester. And this would be given maybe about a third of the way through the class. But realizing that people don't have AP biology, like the students, I'll try to explain things, but if anything's not clear, please raise your hand. I'm happy to explain it to the best that I can. And in addition, if any of you have questions about teaching or this particular class, again, interrupt, raise your hand. I'm happy to answer questions in that vein as well. So I thought this would be kind of fun. So this particular class was designed a number of years ago, and the goal was really to integrate fields that are often taught separately. So, you go to high school, you go to college, you take your chemistry, you take your biology, you take your physics. But of course, in reality, these are not separate entities, and there's a lot of crosstalk between biology, chemistry, and physics. In fact, one of the more fun collaborations that my lab has is with a physicist. And so we wanted to get this idea across to the students starting at this very early stage and not have them thinking that topics were completely separate. So this class attempts to do that. It tries to integrate chemistry into thinking about biology. As you'll see, we try to engage the students in lecture. And that's actually quite a task. You have 500 kids, and we're trying to interact with them. We do it partly through sections, but we also try to have a back and forth with them as much as we can in lecture. We're dealing with a really diverse student body. So we've had kids tell us that not only had they taken advanced placement chemistry and biology, they had then gone on and essentially helped teach it. So they'd taken it in their junior year, helped teach it in their senior year, and now they're in the freshman class. So we have those kids. And then at the opposite extreme, we have kids who've never taken upper level high school chemistry and biology. And we're trying to get all of them to the same point so that as they move on through their years at Harvard, they can all access these different classes equivalently. And finally, the students who come in, most of them have an interest in science. Many of them want to become doctors, but certainly not all of them. And a goal is to give them the kind of background that will help build an informed public, that when they're faced with thinking about medical decisions and health decisions, they have a sense of what goes on behind the scenes to get to the kinds of choices and decisions that are made, that if they're reading the New York Times about a new drug, they'll have a slightly more critical perception of how things work. So that's kind of what we're thinking about. We're going to talk today about this topic here. And it's part of a series. We start by talking about the structure called membranes, and I'll define it for you in a minute. And then we begin to think about how those affect various processes in cells. And we ultimately end up talking about cancer and how a particular drug affects a particular protein that is known to cause a particular blood cancer. And so you can see this is 6 lectures out of the course. We start with very simple chemical and biological principles that ultimately will give us a fairly sophisticated understanding. Unfortunately, I wanted to talk about this to you. I think the chemistry is just too complicated. But I'm happy to talk to anyone about the topic afterwards. So here's where we start. If we're in the lab, in a chemistry lab, we can do lots and lots of reactions, and we can do different reactions, essentially setting up different flasks for different kinds of chemistry. The body needs to do this as well. The body is doing thousands of reactions, and we need to be able to segregate things just as we would segregate different kinds of reactions into a flask. So how do we do that? So on the one hand, we have certainly different organs, many different types of cells that have very particular functions to perform, such as the kidneys for filtering water and liquids. But we can also think about it at the level of an individual cell. So if you remember that bodies are made up of cells, and this is sort of what a cell might look like, cells are not homogeneous, they have all sorts of little structures within them where they can, again, partition reactions that they need to undertake. And where we begin this lecture is thinking about how one constructs different compartments, both to generate a cell and to generate compartments within a cell that will allow cells to perform very specialized functions. And the key to that is what's outlined in black, and that is the thing called the membrane. So for the students, I actually give them a list of what they're going to learn today. And for the students, we actually give them a very clear idea of where we're headed, which they seem to find helpful. Okay, so what is a membrane? And you can think of it as this kind of structure. So this is an artist's rendition of what surrounds your cells and keeps what's inside the cell inside and what's outside outside, and it's all these little pieces here. And it's made up of this. So it's a very heterogeneous molecular composition, but a main building block is a structure like this. And this is called a phospholipid. Has anyone here heard of phospholipids? Great. Oh, wow, you guys are on the ball. This is what it is. It's composed in kind of two sections. This here is a piece that's called nonpolar or hydrophobic. These are essentially long chains of fatty acids, lipids, if you will. And these do not like to interact with water. And then this portion here, called polar or hydrophilic, is a part of this molecule that actually interacts very well with water, shown here. And the reason they have this different ability to interact with water is part of the chemistry that I guess I won't really explain, but these have charges and these do not. So if you take this kind of molecule, and the body and cells are in essence water, so if you throw this into water, what do you think might happen? Separation. Yep. Anyone else? If you know, you don't have to shout it out, just take a guess, yeah? Say what? Exactly. These things form what's known as a bilayer. Bilayer because there are two. So they do this where the portion of the molecule that doesn't want to be near the water is next to other portions, so this keeps it protected from water. And then these sections that I told you want to be near water, or happy to be near water chemically, are against water. And you build this kind of what's called two leaflets. And this, if we extrapolate, might look something like this. So this is sort of that, and this is going to become this. And can you again hazard a guess why? Okay. Say what?
The structure has lower energy.
Okay. It does have lower energy. Any thoughts why that would be? Why do you think this sheet would rather be a sphere than a flat, open sheet? Say what? More surface area? It's probably pretty similar. Yeah?
A lot of it probably has to do with electrostatic charge. Essentially, positives and negatives attracted to one another, so it maximizes the favorable positive and negative attractions.
That's definitely getting on the right track. And in essence, we were talking about this structure here. So the charges, positive and negative charges, that's all these green parts. And water has charges. But this section, as I told you, doesn't want anything to do with charges. And if you think about it, that's what's on these edges. And so if these edges are exposed to water, that's a really bad thing. And so instead, it forms a sphere. and now the black section is nowhere exposed to water. And so you can see just intuitively that by the chemical property of what this phospholipid is, you start to generate what looks like a cell. Okay. So here is a look at a cell. We're looking under the EM, and this is actually membrane. And you can see it's forming a little sphere, just like what we were talking about, where it's separating this from this. These are two parts of the cell. And then you see all these other structures within the cell. This holds the DNA, for example. This does a lot of energy production, and so forth. And so immediately, you have a very simple, energetically favorable way of building different structures within cells. Now, these lipids are interesting in that they're not just all of one kind. They actually come in different shapes. You can have something that looks like this, makes a conical shape, something like this. And if you start putting these together, you can actually build very interesting kinds of structures and shapes that the cell uses to generate different kinds of forms. And so, for example, so what we have here is if you look at these different kinds of structures within the cell in this artist's rendition, by using different flavors of phospholipids, you can begin to generate these different kinds of shapes. Another cool thing about them is that they're very motile. They're not static at all. So if you're looking at this kind of a sheet, they're jiggling around all the time. They're not sitting still. They can move in this plane. They can kind of twist around. The only thing that they don't do is they don't flip sides. So in essence, you start to define one of these leaflets as inside and one of these leaflets as outside. Okay. So, yep?
Is there a fundamental limit on the size of the sphere that it is poured into?
Is there a, what about the size?
Fundamental limit, because the larger sheets would be genetically arrested.
I mean, they certainly, cells will give sort of structural, like you might have the steel holding up a building. Cells do the same thing. They have certain struts to help maybe make something stronger. So that exists. But in terms of the energetics of just that kind of bilayer structure, I don't think there's any limit on that. Okay. So, as I told you, we try to engage the students, and we do it by something called a breakout, where we ask the students to solve a problem. So I'm going to give this to you and see what you guys make of it. And yeah, let's see how this goes. Okay, so here's the question. We're talking about two kinds of fish. One's called C. hamatus, and they have lipids with what's called unsaturated fatty acids. These are fatty acids that look kind of like this, kind of kinked. And then they have a lot of this thing, particular one called phosphatidylethynolamine, which is this here. They also have another fish, this may be too hard, called phosphatidylcholine, which is this nice flat one. And I told you that with the different structures, different structures of phospholipids, you can begin to assemble different kinds of structures as they pack in together. So if you keep that in your head, what do you think? C. hamatus membranes, which have more of this, are more fluid than those of C. auratus, less fluid, or the same? This might be a little hard.
What is fluid?
Fluid is that mobility.
Oh.
So fluid is they're jiggling around like this, and we talked about how they have different shapes here, and these different shapes kind of pack in, just like it's drawn here. So as you pack them in, they move but they're just like a crowd of people kind of jiggling against each other that can influence how much they move. So, given that sort of broad scheme, which of these would you guess? Say what? A?
I would guess A.
You guessing A? Who else thinks that? You think A? Okay. Anyone else? A? What about B? Anyone takers for B? All right, we have some Bs. Looks about 50-50. Anyone thinks it doesn't matter at all? Say what? Yes, I'm just taking the head count. Okay, so who wants to say why they think A? Okay, give it a shot.
If we're saying A, those have more kinks
Yep.
structure, right?
Yep.
That's by the graphic you have on the left.
Yep. And this is the structure of this long name.
So
Yep.
it seems to me that those kinks would give the membrane more structural options than something that was just a straight line.
Okay. You have an idea? No? Okay, we'll come back to you. Other thoughts? Any B proponents want to say why they think B? Yeah.
I think that the kinky ones would kind of get off tangle with each other, and straight ones would have an easier time.
Oh, interesting. So you think the straight ones.
Would flow easier.
Uh-huh. Yeah, I can see that thought. Any other? Yep.
The big words are kind of confusing me, so I like to get them off, but the unsaturated ones can pack closely together, so that would cause them to be less fluid because they'd be so close together.
Because they could come right next to each other. Okay. These are great thoughts. And it's exactly these kinds of discussions that we get into with the students. And in fact, the right answer is shown here. So C. auratus, that's your basic goldfish. I have to say, when I do class, I tried this last year, I give them candy for talking in class. So if I had candy, I would give you a candy bar. This is a goldfish. These are these fish that live in the Antarctic, where it's really, really cold, and they have to deal with the fact that all the processes we think about normally happening at regular temperatures are actually happening in the freezing cold. And one of the ways they do that is to actually build their membranes in a different way. So if you think about butter at different temperatures, makes maybe intuitive sense. You chill things down, they're going to kind of solidify, so you want this to be more fluid. And the way is that things cannot pack as well, and they have an easier time packing when they are flat like this. So I thought your idea was actually a very cool one, but it ends up being the opposite, and I'll show you why. So here's a chemical diagram. This is this very straight molecule, and here's one with a kink. And what happens is when you have a whole solution of these things, they begin to interact with each other. And they actually set up kind of a charge, so that there's actually an attraction between one lipid molecule here and another one here, basically a fat. And so if you think about chemistry, normally chemistry, we don't think of them as having a charge, but they have a little bit of one. And this is what lets them pack in the way we were talking about. And it has a really pronounced effect. So these are two different lipids. They have very similar chemical makeup. For example, they have the same number of carbons, these black pieces here. And the difference is that this is kinked and this isn't. And as a consequence, this, well, let's start here. This one melts at 56 degrees. So it's a solid below 56, and then it turns liquid. This one, on the other hand, because it has all those favorable interactions, it takes much more energy. You have to heat it up a lot more to get it to melt. So that's pretty cool. And the way that plays out in the cell is, whoops. Good question. Saturated is like this. It means there are no double bonds. versus where we have here. So both of these are unsaturated. You may have heard something about cis and trans unsaturated fats. This is cis. It makes this kink. This is trans, and it doesn't. So this, although it's unsaturated, it looks almost like a saturated fat. And this, on the other hand, promotes liquid, kind of a liquid state because of the kinking. So, for example, olive oil has a lot of this kind of thing. Candle wax is much more like this. Is that? Yep.
Can I answer a question?
Why don't we move on and we can get back to it later. Okay. Other questions? Okay. Oh, yeah? Say what? Tastes better? I don't think, excuse me, that the degree, well, I don't know if you guys read about McDonald's today in the New York Times, but there's clearly many people who think these trans fats taste just great. When I was a kid, eating at McDonald's was the most exciting thing ever. But olive oil is pretty good too. So, yeah, I don't think there's one flavor that goes with cis or trans. But health has placed much more emphasis on this kind of configuration. Yeah?
I'm curious, a teaching question. Do you teach the full class in Fahrenheit?
The full course in what?
Fahrenheit, the previous slide you were showing multi-points in Fahrenheit.
At this point, yeah. So for example, in my lab, we work totally in Celsius. But, they're just coming out of high school. They're babies. They're very cute. They're incredibly enthusiastic and pretty nervous. And I remember as an undergraduate myself, you walk in there. I came from a high school where there were maybe 65 of us in my graduating class, and individual classes had maybe 15 kids, very small. And you walk in there, and it's 500 people. It's nuts. And back when I was a student, people weren't on computers. They had those pens that change colors. Does anyone remember those? So the professor would pick up a different colored piece of chalk, and the whole room, you would just hear this click, 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 as everyone changed color. And it was totally intimidating. So I have great empathy with the students as they come in, and this is what they encounter. So we're trying to get them excited about science, learning about science, but not completely intimidated that it's their first semester at Harvard and they're going to have 500 facts thrown at them.
Students are, I think, more American than I thought. I kind of pictured Harvard as extremely international in most countries.
We have kids from all over, but the majority is certainly American. Yeah. Other questions? Okay. All right, so this is your structure. And what we get actually in the cell is that we can have both types of phospholipids. We get, on the one hand, these straight that are kind of more organized, if you like, and we also have these kinked that are tending to be more like this and more fluid. So membranes have both. Okay. So this is just a recap. They have different shapes, they have different saturation, and as a consequence, they tend to be more or less fluid. So they have this propensity, but of course, cells are subject to different conditions, and here are two extremes. I think this is kind of an amazing photo here of this guy. And here's the opposite extreme of Boston in the winter. And this is a problem for cells. And the reason is the following, that because of these characteristics we were just talking about, these membranes have a tendency to go from something quite solid to something quite fluid. And they do what's known as a phase transition, where the transition is really fast. So as you change the temperature, it'll be solid, and then suddenly, boom, it'll be liquid. And I think butter is a really great analogy, actually. So it's, this is all lipids. It's not phospholipids, but it's lipids. And of course, it's solid when you take it out of the fridge, and as you warm it up, it's going to melt. And this would be really bad for cells. You want them at that just fluid, but still intact. So cells are confronted with this problem of how to control this phase transition. And you don't want to be this. Yeah? This? So here, sorry, I didn't explain that very well. We're looking at temperature. So this is the temperature along the x-axis going from low to high. And we're going from a solid state to a fluid state. So at low temperature, you're more solid. Oh, I'm just trying to illustrate that I don't know if there's a particular measurement for more fluid-like, but more to give you the concept that there is this very sharp transition where you go from solid to liquid, and that's what's so hard for a cell.
Kinetic energy or something like that? I mean, some solids have very low kinetic energy, some fluids can have more kinetic energy. So
It's not really kinetic energy, but I don't know if there is. What? Viscosity would be one, yeah, that would be one readout, that's true. Yep. So cells want to be, they don't want small changes here to give these huge shifts here, okay? They don't want to do this. And one way that makes it easier for a cell is if they can change this curve to be kind of flatter, so that changes in the temperature are actually going to have more subtle changes in how the membrane behaves, okay? And the way that they do that in animal cells is this familiar molecule here called cholesterol. So cholesterol is magic. It's a really amazing thing. It's been very much maligned, but it's actually doing a great service to you. And it has this very interesting kind of a structure shown here. There's a portion of cholesterol here at the top that is hydrophilic. It is happy to be next to water. And then the rest of the molecule is very hydrophobic, but it's in two portions. There's this here, which is what we would call a ring structure based on the chemical structure. And then there's this tail region. And all of this is very hydrophobic. So this is much more similar to the portion of the phospholipid that does not want to be juxtaposed to water. And so if you have cholesterol and you add it to the membrane, this is how it will orient. So this polar head will be close to the polar heads of the membrane, and the rest of cholesterol is going to be sitting here near the tails. And within your cells, you have an enormous quantity of cholesterol sitting like this. Cholesterol is the molecule that does this for your cells. And it makes the transitions much less likely to happen. So one thing we go through in this class is showing how the chemical attributes of cholesterol can actually control membrane fluidity. So let's start by thinking about high temperatures. So at high temperatures, it's more likely things will be more liquid, everything can move around more, and the cell would like it to be a little less mobile, a little stiffer. And cholesterol can do that. So it's going to decrease lipid mobility. And it does it in part through this ring structure. The ring structure is very stiff. It's not given to moving very much. And so it actually can block mobility. And it does it not just with itself, but because it has interactions with lipids in the membrane. So here is one lipid in the membrane. Here's cholesterol. And if you look at it kind of intuitively, you can imagine how these two fit together very nicely. And without going into too much gory detail, the head groups here, the water-loving hydrophilic parts, have all kinds of interactions that work very well. And then these nonpolar parts can also interact in very stable ways. And so that allows them to form, in essence, kind of a partnership. And finally, the shapes are very different between the one and the other, and they, again, fit together very well. So that means that when you are looking in a membrane, cholesterol and its ring structure and so forth is interacting with this molecule called sphingolipid, and the two of them are like a pillar that will stop the membrane from moving around so much. Yep?
I just know just from the common top stuff that
Uh-huh.
there's good cholesterol, bad cholesterol.
So
Is there a difference between the ability to stabilize the membrane between good cholesterol and bad cholesterol? What makes something good cholesterol versus bad cholesterol?
that's the packaging. So cholesterol is cholesterol. But in our bodies, it's not just floating free. It's actually bound into particles. And those particles have been correlated with good and bad medical outcomes. But cholesterol itself, this is it.
Oh, so cholesterol is a molecule. It's not like there's cholesterol,
Exactly.
Okay, all right.
Exactly.
What's the F8?
Say what?
What's the F8?
Sorry, it's just a shorthand. It's the fatty acid tail. You know, we looked at those long structures that I showed you. Don't have a lot of charges, and they kind of like to pack together. That's this. This is just the molecular drawing of it. And cholesterol has something similar, and so those two fit against each other quite nicely. Shape and charge and interactions. And so you build this kind of structure, and if you put that in the membrane, it then, it's kind of like I think of it as sort of a pillar that just is going to slow down. So if things are trying to move, and then you have this big rock there, it's going to just slow down the movement. So it's helping the membrane in that way. Okay. Yeah?
OH and NH?
Say what?
What OH and NH?
In the previous, that's the actual chemical nature. Here, for example. So a hydrogen bond is a way that molecules can talk to each other. And it's a type of charged interaction. I think for the purposes of this discussion, don't really need to know about it in gory detail, but know that water is filled with hydrogen bonds and can hydrogen bond with itself and with other molecules. And so the reason that these structures aren't very happy in water is they're not very good at the hydrogen bonds. So water would rather hydrogen bond with itself, that's a lower energy state chemically, than interacting with these. And as a consequence, that's why they partition. So with oil and vinegar, the reason they separate out is water will have hydrogen bonds with other water, or vinegar, same kind of thing. And these, the oily parts, will interact with themselves, but there isn't really a great opportunity for the oily part, which is this, to interact with the sort of vinegary, watery part, which is this, because of this difference in hydrogen bonds.
So in those pairs of letters, H is hydrogen, but what is O and N?
I'm sorry, what?
In those letters. O,
Yeah, these are actual chemical structures where it really helps to have had like chemistry, AP chemistry. I can sit down with the periodic table with you, but I think for here it just seemed a little complicated. So there's a whole series of chemistry, you know, the kind of structure of the atom. So here's chemistry in 2 minutes. Different kinds of entities have a chemical structure where they have electrons, and for whatever reason, you can think of them having shells, and these shells have 8 electrons. So atoms are really happy when they have 8 electrons. And if a atom has 7 electrons, then it's looking around to pick up one more electron. And that's kind of a chemical thing. And that's what drives, for example, certain kind of bonds. If this molecule wants to gain an electron and this atom wants to lose an electron, then they're a perfect match. So table salt, sodium chloride, that's what's going on. You're sort of completing each other by sharing electrons. That's what a hydrogen bond gets to. That's what these get to. But it's a whole course in itself, so I don't think I can really go into it. But that's the long and short of it. Okay. So the other part of things is this tail here. Cholesterol has this long tail. We talked about this at high temperatures. At low temperatures, you now want the whole membrane to be more fluid. And the way that that works is that this tail disrupts the ability of these long chains to pack in. So if you look at this cartoon, you can see kind of intuitively that if two of these very long chains were going to come in together, this is like a little wedge that's going to disrupt that. So depending on the temperature, that tends to favor either the solid side of a membrane or a liquid side of a membrane. Different aspects of cholesterol are more important, and they make this curve, this S curve, much flatter. And that helps the cell out quite a bit. Okay? So the big lesson is you really need cholesterol. For all the discussion there's been about how bad cholesterol can be, it's actually very, very critical for all of the membranes, all of the cells in your body. Okay. So the last thing I was going to touch on, and just very, very briefly, was that there's more to a cell than just the membranes coating it. There's also proteins. And there are lots and lots of proteins embedded in cells that have different functions. And one of the ways that we can know this and see this is illustrated here, where there's a protein called green fluorescent protein, or GFP. And I don't know, some of you may have read about green fluorescent protein. It's very cool. It comes from a jellyfish, looks kind of like this. And if you shine blue light on this protein, it will glow green. And it will glow green even in a living organism. And that means that you can watch a live cell or even a live animal and see what's going on with this GFP. So I thought you might like to see an example here. What the scientists have done is they've taken two proteins that normally sit in this membrane that we've been talking about, and they've tacked on these fluorescent proteins, that came from a jellyfish. And here you can see how these proteins are moving around in the membrane. So you've got the phase of a membrane, you're just looking at it from like the top down, and you can see, and I don't know if you can tell, but I think it's kind of cool, the green is much more mobile than the red. So you get the sense that some parts of the membrane are more static, and other parts are much more liquid. And because of building with different lipids and cholesterol, we can actually generate a structure and then proteins within that structure that behave like that. So in essence, you can think of the behavior of cells that very much depends on the chemical properties of these molecules that are the building blocks. It doesn't require any energy put into it. It is the natural chemistry of those molecules that builds up the structures that the cell uses to generate this membrane. And I thought I would just finally end on a Cambridge note to say one of the reasons that we know so much about cholesterol is from this man here. His name was Conrad Block. He was a Harvard professor for many, many years. And I was looking at this photo, and I'm pretty sure this is Cambridge, although I'm not entirely sure where. Maybe someone will recognize it. He figured out, in large part, how cholesterol is made in our bodies. What are the enzymes that build up those ring and tail and polar groups. And for that, he won a Nobel Prize back in the 1960s. So, okay, I thought I would stop there, and I can answer any more questions. And thanks for coming.
Let's thank Susan. I actually want to take your class now. So if there are any questions, please come up to the microphone so we can get it on.
Well, you can shout out. I can, yeah? I spoke too fast. I can't understand you. Say it again.
Is cholesterol present in all domains of life?
It's not in plants, but they have something very similar. So they have a related kind of a structural protein, but not cholesterol itself. Same thing. So cholesterol is in animals. So your pet dog or cat, C. elegans, Drosophila, so forth, they all have cholesterol in their membranes.
Can I ask one question? First of all, this is kind of what I do for a living. I actually taught AP chemistry at a Catholic school. I was very impressed by what she presented. I was actually took class in this hall in 1997, biochemistry. I studied with Stu Schreiber and people like that. I guess my question to you is, I'm sort of assessing the university and seeing how it's changed a little bit from 1997 to today. That's a better lecture than what I got. So I was very impressed by that. You know, the knock that I hear on the street about Harvard is, this is the place you go if you're already a graduate student when you come in, because it's about research. It's the pedagogy is such that, you know, we love your recommendations by your students, but you get paid to publish or perish.
That's the faculty.
Yeah, the faculty.
On
that this is a publish or perish, not a teach and preach. I don't necessarily agree with that. I can think of people that I had here who were great professors, but how would you respond to that particular challenge of Harvard?
I heard that same comment. When I was here in the 80s, I think it was true, or much truer. There were many fewer courses, and some faculty were very much vested in teaching, but others were less so. And there wasn't really the emphasis on teaching that we have today. Today, I think it's very different flavor. And Harvard as a whole is very interested in teaching. We all teach. The most famous, lauded faculty here are teaching, as are the assistant professors. It's not like you can get out of it because you are famous or traveling a lot or whatever. And in fact, I would say often the really famous professors, a lot of them are very good teachers. They're just very good, very articulate. They're good communicators. You know, like Conrad Block, who taught me biochemistry, you know, but he was also a Nobel laureate. We have a lot more courses than we did when I was in college. So I think that whole flavor has really changed, and there's a big interest. And of course, I find it's a real pleasure to teach the students. They're very enthusiastic and just very energized. They're excited to be here. And despite some of them looking at Facebook in the back of the room, they really want to understand the material, and they're pretty engaged. So I think we're very lucky in that regard to have them as the students. And we do a lot for them. I mean, we have the lectures, and we have sections, and we have office hours, and you know, there are many, many activities where they can interact with faculty.
What are the mechanisms for ion transport across the membrane? You were going to talk about that.
I'm not, because it's complicated, but there are special proteins that sit in the membrane and allow ions to pass. And one of
They come against concentration gradients.
they will go against, yeah. It's actually a remarkable feat. They move at the limit of diffusion. They can go so fast. So for example, we in class go through a potassium channel, which is a kind of a salt. It's really critical for all cells, and it's also very critical for nerves. It's how nerves talk to each other, and that's how certain salts move back and forth across the membrane. And the proteins that do it have exquisite specificity where they're getting the wrong salt, the wrong ion, 1 in 1,000 maybe, and they're doing that at the speed of diffusion. So it's a really remarkable and beautiful example. And
So it's protein mediated?
it's protein mediated, and the protein has set it up where it's just pinging these things through at an incredible rate.
And what's the advantage? Is there some advantage to the cell being fluid?
You mean the membrane?
The membrane.
Yeah, I mean, I think if it was rock hard, that would probably be a problem. It's sort of deformable. You know, you can, if you bring a steak home, you can push it around without it falling to pieces. And membranes are part of that. Things move and diffuse, and there are structures probably built up in membranes. It's kind of controversial, but the thinking is that, you know, two proteins come together and have an interaction in the membrane and then come apart. And for that, you need the fluidity that things can move in that two-dimensional plane. So, you had a question?
Thank you, Professor. When teaching a class to the general public, especially a college-age public, how separately do you think about the two concerns of, one, actually teaching the content of science, and separate from that, getting people interested enough in the topic to be motivated to learn in the future? And as a follow-up to that, do you think it's possible to inspire future curiosity separate from some threshold of concrete understanding in the present?
Yeah. So, you know, do we just pack them full of facts, or how do you move beyond that kind of thing?
I don't mean this in a critical thinking sense.
Yeah. So I would say we do it in pieces over years. So what we find in this freshman course is many of the students come in, they've taken the advanced placement courses, and they know a lot of words. They have a lot of terms, and they think they know everything, and in fact they know very, very little. So they will throw certain terms at you, and if you push them at all and ask them about it, it's clear they don't really understand it. So our goal at this point is to sort of get them past that and begin to talk about what certain things really mean, and also, as I say, form this baseline, and to get them excited about how you can begin with these chemical principles and then get to biology. They are really excited about that. For example, in the 6th lecture, you know, I show a structure of a drug, how it binds to a particular protein, why that affects, why it can kill the cancer, what that actually does, how you get resistance to that drug, and how you rebuild it. And I think for them, seeing the chemistry and what that means in real life is just very, very exciting. In the sophomore year, for the students at least who are taking biology, we do a lot more about how you get to answers. So instead of just telling them, for example, that a membrane has this behavior or cholesterol has this behavior, we talk a lot about experiments that led scientists to those conclusions, so that they begin to get a sense of, you know, how do you get to answers, and what kind of questions might you ask, and can you begin to think, be more involved in the sort of intellectual process. And a lot of them join labs, and will actually try to do some research. So I think it's kind of in stages that they come in pretty curious. You know, we're lucky in that we get a very excellent group of students, so they're not passive at all, they're very engaged. But our hope is that we can deepen their thinking process, and their whole approach to science, that we can get them away from just thinking about how to get an A and how to score well on a quiz, and have them think more about kind of really learning and thinking through the processes and understanding this relation between, biological behaviors and the chemistry behind it. And so we are trying to change how they approach courses.
For a long time, we were told that food with high cholesterol is not good for health. And recently, they changed the guidelines. It's okay to eat butter or eggs. And you say you should eat your cholesterol. So what's your take?
What's my take? So my first...
May I ask a related question? High density and low density. What are the proteins? What are they dense with? And if high means more and low means less, why is it better to, if that's the case, then, and what is it dense with, and why is it better to be more dense with what?
So let me say two things. First of all, I am not an MD. Like, I can't deal with blood at all. I would be the worst doctor in the world. So that's the caveat here. HDL and LDL is very much about how cholesterol is packaged and moved around in your body. It's not about this structure here, this cholesterol, and it's not about cholesterol in cells. And I think cholesterol got a really bad name. And so people thought, cholesterol is terrible. In fact, it's essential, right? Everybody needs cholesterol because it's this huge component in your cells. And so the thinking was that it can build up in your body. People are much more mellow about cholesterol now. It's much hard, my understanding, it's very difficult to lower cholesterol levels because your dietary intake doesn't have that big an effect. So it really is, you really have to have like no cholesterol if you want to drop it. So, and fats in general. The interest in fat and thinking fat caused obesity was in part very much, there was one person who really believed in it, in large part because there were these population studies and looking in part at the Japanese diet and arguing, saying, okay, well, the Japanese don't get a lot of heart disease and Americans get more heart disease. If we look at the difference, well, Japanese don't eat much fat, so that must be the reason. And they had our interests at heart, but of course there are lots of differences between a Japanese diet and an American diet, and it was really jumping the gun. And now people are starting to study salt, fat, cholesterol, and it's not nearly so clear that those ideas back in the 40s and 50s are in fact true once you actually do a controlled experiment. So I love eggs, I eat them.
Hey, what happens if you take all the cholesterol out of a cell?
Out of a cell. I don't think anyone's done that.
Oh, let's do that. I think that, let's do that. How would you do it?
Well, in worms, the organism I study, which is a small worm, it gets cholesterol, it can't make it gets it from the soil. But and if you do that, they are not happy and they end up being sterile, so that's not good. And then they basically die. But cholesterol has another role. This one is, most of the cholesterol. It's also used for cell-cell communication. It's attached to certain proteins and then used for cells to talk to each other. And that's another role. So if you got rid of all cholesterol, which you could never really do, you would be you'd have severe problems because of that.
So I want to thank Susan, and I want you to come down afterwards if you have more questions. I think it would be a perfect time. That was a great talk. Thank you very much.